Yeah, so I will present this new method, which is based on mechanistic principles, and it can be used to study dynamically evolving regulatory networks. And first, a few words about the background. So basically, if we consider or observe any biochemical system, it's obvious that there can be many interactions between the molecules or mechanisms that might be affected by latent mechanisms that are not observed. And it might lead to this kind of rewiring of the interactions that we are observing. And in the context of transcriptional regulatory networks, these latent effects can be like transient stimulation or transiently changing signaling pathway activities, or epigenetic state, which is changing over time. So basically, the challenge is to detect if this kind of rewiring occurs in the system of interest. And on the other hand, it would be also very nice that we can also do some kind of network inference in the case of rewiring. And for this purpose, we have developed the so-called latent effect mechanistic model, which combines traditional ODE modeling with the latent component that mimics the rewiring effects and thus makes the model class more flexible. And in addition to this new model class, we also introduce statistical framework that can be used to calibrate the model structure and parameterization in a data-driven manner using time course measurements. Before going to the formal definition of the model, I will present the modeling idea through this simple toy example. So we can consider a gene which is activated by a transcription factor A. And once the gene is activated, we obtain the gene product B. And this simple biochemical mechanism can be summarized using this network where we have the interaction between A and B. And further, it's rather straightforward to construct the corresponding ODE model where we have the degradation mechanisms, basal activation, and then the component corresponding to the interaction between these two molecules. The situation is a bit different if the gene's locus is covered with some kind of silencing marks in the initial state. And then once the cells are treated with some drug or some stimulus, then there can be some unknown enzymatic signals that remove the silencing marks, and eventually we reach the active state where the transcription factor can bind and we obtain the gene product. So mathematically, this means that we initially have this kind of network where we don't have the interaction between these two molecules, and then we switch to another network where this interaction is present. And typically, we observe cells on population level, and because molecular reactions are stochastic in nature, the cells in a population don't switch to another phase synchronously, and thus we have a population that is gradually moving towards the other phase. And this movement can be described using this kind of latent process, where the blue component corresponds to the phase where we don't have the interaction between the molecules, and the red corresponds to the activated phase. And here's the formulas that can be used to generate this latent process, and depending on the parameterization, the switching can be rapid or it can be very smooth. And now when we have the traditional ODE model, we can construct the latent effect mechanistic model by coupling the latent process with the ODE model. And the coupling is done by using this kind of weighting function, which is here coupled with the functional component which corresponds to the interaction between A and B. And this weighting function has two binary parameters, Z1 and Z2, and they are actually the parameters that determine the structure of the model. So if both of them are 0, we don't have interaction at all. With this configuration, we can have early interaction. With this configuration, late interaction. Or if both of them are 1, we have one sustained interaction, and this model then corresponds to the standard ODE model. And in this toy example, we have coupled the latent process with the ODE model only for one mechanism, but in general case, we could do the coupling, add these binary parameters, and add the coupling also for degradation processes and the basal activation. So in general, LEM model can be constructed using any standard ODE model and coupling it with the meaningful latent process. And the latent process is always depending, or each individual weighting function is depending on a set of binary variables that determine the latent states that are combined with the certain functional components. And in a similar manner as in the toy example, the model structure depends on these binary parameters. And in our work, we have noticed that it's very convenient to collect these binary parameters to this kind of matrix, where each row corresponds to one interaction or mechanism, and then each column determines the network structure which is active in a certain latent state. And this results in a very flexible model class, but on the other hand, we have a huge number of different configurations for this Z matrix. So if we have J interactions and M latent states, we will have 2 to the power of J times M alternative models. This basically leads to a model selection problem, and we carry out the model selection within Bayesian framework, which means that we are essentially interested in this posterior distribution over the different configurations of the Z matrix. And we approximate the model probabilities by using this expression, which is basically based on Bayesian information criterion approximation for the marginal likelihood. Okay, now I have pretty much described the method, and next I will outline how we have applied this approach to study TH17 cell differentiation and rewiring during that process. So we know from previous studies that this kind of rewiring occurs during early phase of TH17 cell differentiation. And we also know from these studies that there's like sequential phases. So there can be early interactions, sustained interactions, and late interactions. And this prior information is like something that we can use to construct a latent process that we use in our model. So in this latent process, we have 3 phases, and we have 2 parameters, tau1 and tau2, which determine the time points when the switching between different phases occurs. And we also have parameters that determine how smooth these transitions are. So they can be very rapid or very smooth. And here is the parametric form of the latent process, and here are the parameters that are also then optimized during the calibration. Then we have also prior information about the core genes that drive Th17 cell differentiation, so we concentrate on these 5 genes. And we also have collected from literature all possible mechanistic interactions between these core genes. So there can be this kind of activating mechanisms, inhibitory mechanisms, synergistic activations, and we also allow basal activation and degradation. And here's the formula for the standard ODE model that can be constructed for the full network containing all possible mechanistic interactions. And here we have basal terms, linear terms for activating links, then nonlinear terms for synergistic activation and inhibition, and then degradation. And as I mentioned earlier, in order to obtain the LEM model, this model is then coupled with the latent process that I introduced. We use RNA sequencing data, time course RNA sequencing data, to inform the model, and to combine the model with this data, we use Gaussian likelihood, and we define relatively uninformative prior over the non-zero elements in the Z matrix. And this prior is defined in order to penalize the more complicated models, that is like added ones to the matrix in the case when the actual parameter dimension remains constant. But it's fairly uninformative. Then we use forward-backward stepwise search algorithm to explore the model space, and it simply means that we start from empty Z matrix and flip the elements in the matrix as long as we get improvement in the model fit. Okay, and here are the subnetworks that we predict by calibrating the model. So there are 3 subnetworks which correspond by color coding to the predicted latent process. It is clear that we have certain time windows where different network structures are active, but on the other hand there's also overlapping behavior because these are rather smooth, these transitions. And it is known that STAT3 has a central role in early phase of the Th17 cell differentiation, and thus all these predicted network structures have also biological interpretation. Then we can also observe the predicted mRNA dynamics against the data, which is shown here by red circles, and we see that it's in a very good agreement with the dynamics that we see in the real data. Then I could summarize the whole study. So we have developed a general mechanistic analysis method that can be used to study dynamically evolving molecular networks. We have applied the method to study Th17 cell differentiation, and the predicted rewiring behavior is in agreement with the previously reported behavior. And all in all, the results are biologically interpretable In the future, it would be very nice to improve the search algorithm over the model space so that we could also apply this method to larger data sets. And here are my co-authors and my funding sources. And thank you.